Good morning students. Today I am going to discuss about cell-mediated immunity, a very important topic. Before going into that, we all know immunity is divided into natural or innate immunity and acquired immunity. Natural immunity is present since birth. So acquired immunity, when any foreign antigen enters or when any foreign bacteria or virus enters from outside the body, acquired immunity is produced. And we all know acquired immunity is divided into cell-mediated and humoral. And the topic of discussion today is cell-mediated immunity. So to make it easy, before going into discussion, I want you to differentiate between cell-mediated and humoral. Cell-mediated immunity is T lymphocyte, humoral immunity is mediated by B lymphocyte, and we all know T for thymus. Those T lymphocytes are being produced, processed. Diversity of T lymphocytes all occurs in thymus. That's why T for T, thymus. Same thing B lymphocytes, bone marrow. So we all know that. And cell-mediated immunity, no antibodies are produced. Or humoral immunity, antibodies are produced to kill the antigen. Another difference we know, cell-mediated immunity, MHC is required. MHC means major histocompatibility complex, divided into 1 and 2. I will be explaining that. Here no major histocompatibility complex is required. For cell-mediated immunity, the topic of discussion today, I require antigen presenting cell. So some cells has to present the antigen, then only cell-mediated immunity by T lymphocyte is possible. But for humoral immunity, free antigen, note it down, free, nothing is needed. The antigen can be circulating freely. I need not have any antigen presenting cell to present the antigen. Antigen can circulate freely anywhere in blood or in plasma, in the blood, in any fluids, and that can be taken by B lymphocyte. Why MHC is required is antigen presenting cell. I will write it. Required for cell, or otherwise for cell-mediated immunity, I need an antigen presenting cell. That's why it is called cell-mediated. Yeah, C for cell. Why it is called cell-mediated immunity? I require a cell which is antigen presenting cell. And here humoral, no antigen presenting cell. Very, very easy. No antigen present. So antigen presenting cell can be macrophage. It can be dendritic cell. Note it down. They will ask what are common antigen presenting cells. In general, antigen presenting cells can be macrophage, dendritic cell, even B lymphocyte. B lymphocyte also can present antigen. This is especially for helper T cell. So all these three, macrophage, dendritic cells, B cells, all these can present the antigen to usually helper T cells, and all these three in common as MHC2. Please note it down. I will be explaining. So MHC2, major histocompatibility complex 2. That means MHC1 is there, MHC2 is there. MHC2 is for helper T cells, and this MHC2 is present in 3 antigen presenting cells like B lymphocyte, macrophage, dendritic cells. If it is cytotoxic T cell, again AP antigen presenting cell is needed, which can be all cells except RBC. For cytotoxic T cell, one type of T cell, it is MHC1. And this MHC1 is present in MHC1, major histocompatibility complex 1, which is for presenting the antigen to cytotoxic T cell, and this is present in all cells. MHC1 is present in all cells, which includes all these also, except RBC. Yeah, except RBC, because MHC requires nucleus and all cells contains nucleus except RBC. That's the reason. So another MCQ, MHC1 is present in all cells except RBC. MHC1, which T cell? Cytotoxic T cell. MHC2 is present in macrophages, dendritic cells, B cells. MHC2 is for helper T cell. Okay, now the question, why humoral immunity name? Why that B lymphocyte immunity is called as humoral immunity? Humor means fluid. As I mentioned, free antigen, it can be circulating anywhere. So this B lymphocyte can take antigens from the blood. The B lymphocytes can take antigen from synovial fluid. The B lymphocytes can take antigen from any fluid. That's why the name given is humoral immunity. So I now made very simple. Why the name cell-mediated immunity, which requires antigen presenting cell? Why the name B lymphocyte? Why the name humoral immunity? Because it is fluid. Another important thing, cell-mediated. So it is against intracellular pathogens, note it down. Whereas humoral immunity is mainly against extracellular, that difference also you should note it down, extracellular pathogens. Extracellular pathogens, sometimes the cell itself will be infected usually. For example, virus infected cell or a fungus infected cell, that can be engulfed, the entire cell can be engulfed by macrophage. This is the macrophage and this is antigen presenting cell. So just concentrate what this macrophage does. Macrophage is an antigen presenting cell. It takes the virus which is infected, the fungus which is infected, and this thing is present in either virus or fungi. I am taking example. Now what happens? This will be broken down into various fragments of antigen, and that antigen will be coming to, antigen is coming here, and this is MHC1. MHC1 means what T cell? Cytotoxic T cell. Just to concentrate, cell mediated immunity is against intracellular. So within the cell, the virus is there, and that virus is broken down into antigens. That antigen is brought to the T cell. Here I am taking cytotoxic T cell. So I already told. Please note it down again here. MHC1, MHC2. How to remember? MHC1 is for cytotoxic T cell, killer cells. MHC2 is for helper T cell. MHC2 H. So from 2 I can make H, from 1 I cannot make H. For remembering I am telling. So MHC2, major histocompatibility complex type 2 is for helper T cell, H for helper. MHC1 is for cytotoxic T cell, you can note it down. Cell mediated immunity is against intracellular pathogens. Humoral immunity is free antigen which is outside the cell. So extracellular pathogens. T cells take antigen from antigen presenting cell, no doubt in that. And I told helper T cell is there, cytotoxic T cell is there. Cytotoxic T cell can take antigen only from MHC1. MHC1 cytotoxic T cell. Helper T cell MHC2. So I told 2H. MHC2H. Now just to concentrate. MHC1 is usually expressed in infected antigen presenting cell. MHC2 is usually present in normal antigen presenting cell. That point you can note it down. Sometimes they will ask difference between MHC1 and MHC2. MHC2 you know helper T cell. MHC1 is cytotoxic T cell. And another thing, usually MHC1 is expressed in infected antigen presenting cell. MHC1 usually, if the cell is infected, that is going to be destroyed by cytotoxic T cell through, I will explain that, MHC1. If, for example here some, cell mediated immunity also against intracellular pathogens like virus, fungi, note it down. Also some bacteria, intracellular bacteria like Mycobacterium tuberculosis, Mycobacterium brucellosis and Mycobacterium leprae. Note it down. All these comes under cell mediated immunity. Again repeat, cell mediated immunity is against intracellular pathogens within the cell. That can be either virus or fungi. If that virus, fungi is infected, the antigen presenting cell is infected. If that cell, if the antigen presenting cell itself is infected, means MHC1 cytotoxic T cells. If the macrophage, for example, this is macrophage. The macrophage is normal and Mycobacterium tuberculosis, TB bacteria, which is infected. From this TB bacteria, the antigen is coming here. This is TB antigen from affected Mycobacterium tuberculosis bacteria. From that bacteria, this antigen is entering inside. So now this is antigen, not the entire bacteria. Now this antigen will be taken by MHC2. That's what I want you to tell. Infected cell means MHC1. The antigen is entering inside the cell, but this is normal macrophage. This is infected antigen. This is normal. So, this will be taken by normal antigen presenting cell which is containing the antigen, example TB antigen. That antigen will be carried by MHC2 and which will be given to helper T cell. Note it down. So, this is like a cycle. MHC1 cytotoxic T cell, MHC2 helper T cell. MHC1 is usually infected antigen presenting cell, MHC2 is usually normal. So, now I am going to functions of T cell. Yes, cell mediated immunity. I am explaining that helper T cell, cytotoxic T cell, all those things helps in cell mediated immunity. And helper T cell will help B cell to produce antibodies. Helper T cell will help cytotoxic T cell which will kill. I will tell that mechanism, how cytotoxic T cell is going to kill the organism. And helper T cell will help get the immunity. Suppressor T cell. Suppressor T cells are also called as regulatory T cells. T cells also helps in graft rejection, another function. Fourth one, type 4 hypersensitivity reaction. Note it down. There are 4 types of hypersensitivity reaction, type 1, type 2, type 3, type 4. Type 4 hypersensitivity reaction is cell mediated immunity, another function of T cell. And this type 4 we call it as delayed type hypersensitivity reaction. And another function is usually fights against cancer. Cell mediated immunity, functions of cell mediated immunity is fighting against cancer or kill the cancer organisms or prevent the overgrowth of cancer organism. Fights against virus, fungi, intracellular bacteria. I explained Mycobacterium tuberculosis, Mycobacterium brucellosis, Mycobacterium leprae, and it also fights against cancer. So, all these are, this slide is showing functions of T cells. Yeah, now we are entering into major topic, mechanism of cell mediated immunity. Please concentrate. So, this is antigen presenting cell. So, this is antigen. So, this is MHC2. So, this is antigen. Now, just concentrate what I have drawn. Antigen presenting cell. This is MHC, major histocompatibility complex 2. Usually, MHC2 is present inside antigen presenting cell, and this MHC2 function is presenting the antigen. Note it down. I will separately put a video on major histocompatibility complex. As the name suggests, histocompatibility, checking the, for example, thumbprint. My thumbprint is different from another people's thumbprint. The reason is my MHC is different. Is major histo means tissue. Compatibility of the each one's tissues. So, that is separate entity. Major histocompatibility complex. Regarding MHC, I will put a separate video. Today we are discussing on cell mediated immunity. So, I have drawn an antigen presenting cell which is containing the antigen being processed. A nucleus will be there. Nucleus. Lysosome will be there. Ribosome is there. So, through this process within this antigen presenting cell, the bacteria or virus which are centered inside the cell has been processed into antigen, and this antigen is being presented. See here, MHC2 is presenting. So, first step, antigen presentation. Note it down. Already we told cell mediated immunity means I need a cell which presents antigen, and that, for example, I will put this as macrophage, no problem. Macrophage has processed virus or fungi and the antigen is being produced. That antigen is being presented by MHC2. Yeah, now you have to tell me helper T cell. I can't put cytotoxic T cell here because MHC2 is presenting the antigen. Hope you understood. So, MHC2 H helper H. So, H2, very simple. So, MHC2 is being presenting the antigen and MHC2 is present on the surface of, note it down, MHC2 major histocompatibility 2 is present on surface of antigen presenting cell. So this is TCR. So T cell receptor. So T cell receptors, as the name suggests, TCR. T cell receptor is present on the surface of the T cells. So here helper T cell receptor. Now note it down, antigen is combining with T cell receptor. So for antigen presentation, I require MHC and APC. So first step is antigen presentation. Second step is antigen processing. For example, imagine, this is bacteria and processing goes and antigen is being released. This is antigen. If it is free antigen, that can be taken by antibody and antibody antigen complex. But here the antigen is entered into the antigen presenting cell. Now no other role. T cell only has to do this function. See, again you see here, concentrate here. Bacteria, this is some cell which is doing the breakdown of the bacteria into various antigens. If the antigen is free, circulating, humoral, if the antigen is humoral means oral, fluid. If the antigen is free, that can be taken by antibodies which is coming from B cells. So B cells differentiate into antibody and this antibody is going to kill this antigen, or this antigen produces whatever antibody is needed. But this antigen entered into the antigen presenting cell. Once APC comes, antigen presenting cell means it is T cell. Since it is MHC2, it is helper T cell. If it is MHC1, it will be cytotoxic T cell. That difference you will note it down. Now wait, we will go step by step. Now very important. Antigen is being taken by helper T cell. Now T cell will not be activated. T cell activation has to take place. For that, this helper T cell contains CD4. So CD4 is a glycoprotein. Please note it down. CD means CD, cluster of differentiation. It is very important. It is usually made CD1, CD2. It is based upon the order of invention or whatever it is. The CD, for example, we will tell CD4 count is decreased in human immunodeficiency virus. So CD4 is nothing but CDE means cluster of differentiation, and usually CDs are present in the surface of T cells, or CD count, or CD1, CD2, CD4, CD8, CD28, whatever we tell, CD are concerned with immunity. That you remember. Whenever CD count is decreased, that means your immunity is not good or your immunity is decreased. That is what I want to tell, the importance of CD. So here I am starting with CD4. Note it down. CD4 is present, is a glycoprotein present on the CD4. I will write it here. CD4 is present on surface of helper T cell. Note it down. Now the function of CD4 is, only if the CD4 is present there, this combination which has occurred is being checked. C for check, you can note it down. C check. C for cluster, but for understanding I am telling what the CD4 does. CD4 is present on surface of helper T cell, and the CD4 checks whether the antigen has come with MHC. What I told, T cell will not recognize free antigens. T cell will not take free antigen. T cell will take only antigen coming from antigen presenting cell. Here it is macrophage. Now CD is confirming. CD4 is confirming that it is coming from. Now T cell will not be activated. Now what happens? Again another step is occurring. See the arrow mark? Yes. So interleukin 1. Note it down. IL1. IL means interleukin. Interleukin means connection between two leukocytes. T cell is a WBC, leukocyte. Macrophage or monocyte, modified monocyte is converted to macrophage. So monocyte is a leukocyte, B cell is a leukocyte. So the connection between the two we call it as usually interleukins. So here, since I am mentioning for first time, this interleukin 1 is coming from the antigen presenting cell and a signal is being given. So interleukin 1 is coming from the antigen presenting cell and signals is being given to the helper T cell to check whether CD4 is there, whether the MH double check occurs. Note it down. Double check occurs. So CD4 is present. Yes, CD4 is present. whether MHC2 is coming with the antigen. Yes, MHC2 is coming with the antigen. Whether T cell receptor is there. Yes, T cell receptor is there. Whether antigen binds with receptor. So all these things are being checked by interleukin 1. So they check the connection. Interleukin is a connection between two leukocytes. Again, T cell will not be activated. So now what? So this is first step, CD4. Interleukin 1 is second step. Now third step is CD28 and B7. So this is third. So please concentrate. All these I am telling for T cell activation. So antigen presenting cell is presenting and processing, or processed antigen is being presented. I will make it simple. The processed antigen is being presented from macrophage. For this T cell, why I am putting helper T cell? Because the one which is giving the antigen is MHC2, 2 for helper T cell, 1 for cytotoxic T cell. Okay. Now for T cell activation, first thing is CD4, which is checking that this MHC is coming with antigen or not, or antigen is free or coming with MHC. Second one is, please note it down, interleukin 1 coming from macrophage or APC. Interleukin 1 is secreted from APC, antigen presenting cell. Now third one is, I want you to memorize this, CD28B7 complex. So CD28 is present on, CD28 is also a glycoprotein, cluster of differentiation. CD28 is present on surface of helper T cell, and the CD28 is sending signals to B7. So B7 is present on APC, antigen presenting cell. Again, the CD28B7 is also confirming this combination of helper T cell and antigen presenting cell. So three things are required for T cell activation. Even if one is, if CD4 is not there, T cell will not be activated. Further step, no further step. The T cell is not activated means T cell is not going to function. Immunity is lost. That's why if CD4 count is less means you are having immunodeficiency. Okay, CD4 is present. If interleukin 1 is not proper, for example, interleukin 1 is there, it is not proper, it is decreased. That means this step is affected. The checking of signals from antigen presenting cell to helper T cell is affected. For example, fever. In interleukin fever, I need more immunity to protect against infection. So interleukin 1 usually will be more. Interleukin's secretion is going to be more in fever or any infective conditions. For example, I am telling. So interleukin 1, if that is not there, again problem, my fever will not be controlled or my immunity is decreased. CD28B7, another check. So triple check. CD4 is first check. To check this combo, I will put that combo pack. Yeah, combo is antigen presenting cell with MHC2 coming to T cell receptor of helper T cell. So please note down the combo: antigen presenting cell, MHC2, antigen, helper T cell receptor. So this is being checked by CD4 present on the surface of T cell, CD28 present on the surface of T cell. CD28 communicates with B7 present on surface of antigen presenting cell. Interleukin 1 is coming from antigen presenting cell. I have written here very clearly. So all these should occur for finishing my activation. So now all this has been checked. T cells is activated. Here I have taken helper T cell. So far I have explained this: antigen presenting cell, helper T cell, MHC2, CD4. That's why note it down. CD4 helper T cell is also called as CD4 T cell. If they ask CD4 plus T cell, it is helper T cell. Don't worry. I will explain CD8 later. CD8 T cell means cytotoxic T cell. CD4 T cell is nothing but helper T cell. Note it down. The CD28B7 is known as co-stimulation. So if they ask what is co-stimulation, it is the connection between CD28 from helper T cell and B7 from antigen presenting cell. So this communication between these two is called a co-stimulation, and this T cell receptor MHC2 checked by CD4 is called a co-receptor. So again 2 terms I am telling. Co-receptor is CD4 checking the receptor. Co-stimulation is CD28B7. Okay. So and first step was antigen processing, presentation, activation. For activation only I require CD4, interleukin 1, CD28B7 combo. Yeah. Now what happens? This helper T cell is activated. What this activated T cell does, activated, it secretes interleukin 2. Next step, this activated helper T cell secretes interleukin 2 and this interleukin 2 again stimulates. So this is like a cycle. This helper T cell is stimulated by interleukin 2. That interleukin 2 is secreted from the same helper T cell. So this is plus, coming from this plus. So now what happens? This is still activation, or this can be proliferation or clone. Clone means same type. Proliferation of T cells takes place, and remember all these T cells are specific for that antigen. For example, I will put another antigen. This is black antigen, for example. Yeah, here I will put pink antigen. So all these are black. Now I am putting clones of T cells here. Just concentrate. All these T cells are having the T cell receptor specific for this antigen, black antigen. That's how this is being processed. Note it down. Each T cell has a receptor specific for particular antigen. In the thymus itself, I told no thymus function. Apart from producing T lymphocytes, it also processes the T lymphocytes, and diversity of T lymphocytes occurs in thymus. So thymus is the place where this T lymphocyte is already being prepared for this black antigen only. If this antigen comes, maybe I will draw another T lymphocyte here for understanding. Yeah, helper T cell. Yeah. For example, this is antigen presenting cell. Instead of black antigen, if this antigen is there, this helper T cell will not come. This helper T cell will come because this helper T cell is specific for this pink antigen or violet antigen. So note it down. Each T cell is specific for particular antigen. Don't write black, violet. For understanding I am telling. Now what happens? This helper T cell has been multiplied into many T cells for black antigen. The reason is interleukin 2. So interleukin 2 helps in autocrine stimulation. For this clone to occur, I require interleukin 2 and all these steps. Another function of interleukin 2, please write, activates macrophage. So functions of interleukin 2 is coming from helper T cells, and this interleukin 2 is again going to stimulate the helper T cell. Engirundo varudho, ange it is going to stimulate autocrine. And apart from that, interleukin 2 also activates macrophage. So now we got an idea how the proliferation of same type of T cells to multiply into clone, and then again differentiation of this takes place. So activation, proliferation, differentiation. I have discussed about T cells. Mainly I have discussed about helper T cells. Helper T cells will help other T cells. Helper T cells will help cytotoxic T cells to kill the cells, which I will be explaining. And helper T cell will help B cells to produce antibodies, or B cells which are being helped by the helper T cell. That B cells will be differentiated to plasma cells, and plasma cells will secrete antibodies. All those things we know. B cells will be acting against free antigen. Helper T cell only if antigen is presented with MHC. Yeah, helper T cells type Th1, Th2, Th17. Regulatory helper T cells are there. Suppressor T cells. Suppressor comes for common. This slide I am discussing about helper T cells. So helper T cells are differentiated into Th1 helper T cell, Th2 helper T cell, Th17 helper T cell. Th1 is mainly for intracellular, virus, fungi, or like almost like Th1 is going to help cytotoxic T cell. Th1 is helper T cell, one type of helper T cell which is going to help cytotoxic T cell, and this is secreting interleukin 2, and this interleukin 2 is helping cytotoxic T cell. And apart from that, the Th1 is also secreting interferon gamma, and interferon gamma to help macrophage. We are now discussing cell mediated immunity. So far I discussed types of immunity, and in cell mediated immunity how to differentiate why it is cell mediated immunity, why it is humoral immunity. Th1 is a type of helper T cell which is helping in killing, helping the cytotoxic T cells by secreting interleukin 2. Note it down. I already told interleukin 2 functions. One function is autocrine stimulation. Function I told is it activates macrophage. Interleukin 2 also activates macrophage. and interferon gamma also activates macrophage, that you can note it down. So Th1 is helping in activating cytotoxic T cell via interleukin 2. It helps in activating macrophage via interferon gamma as well as interleukin 2. And Th1 helper T cells, Th1 also helps in virus inactivation. It also helps in cancer cells. Yeah, cancer cells cytotoxic, virus also cytotoxic. Ultimately Th1 is helping cytotoxic T cell. Now coming to Th2, another type of helper T cell is going to help B cell. Yeah, this will be easy. Cytotoxic T cell Th1, B cell Th2. I am making it easy. Here the interleukin is interleukin 4, interleukin 5. You can note it down. Interleukin 4 is for B cell growth factor, which I will be explaining in another video, humoral immunity, which is mediated by B cell. So helper T cells will secrete interleukin 4, which is helping in B cell growth factor. Helper T cells will secrete interleukin 5, which is helping in B cell differentiation factor. If they ask which helper T cell is helping B cell, means Th2. Which helper T cell is helping cytotoxic T cell, means Th1. That you remember. Th1 interleukin 2, Th2 interleukin 4, interleukin 5. Interleukin 4 is helping B cell growth factor. Interleukin 5 is helping B cell differentiation factor, and both these is obviously going to help B cells, plasma cells, and plasma cells ultimately is going to produce antibodies, and all this is being occurring only with the help of helper T cell. Th17 is usually for fungal, bacterial. So I don't want to go in much detail. Helper T cell Th17. Apart from that, regulatory cells, it regulates the function of all these helper cells. Suppressor T cells are present in both cytotoxic as well as helper cells. It is like a homeostasis. Suppressor T cell. For example, if the suppressor T cells are not present, then what will happen? If that may be killing the normal cells also if you don't have suppressor T cells. And another cell, memory T cells. Memory cells are also present for B cells. When your body is exposed to the same antigen second time, the immunity is greater, or when the same antigen is exposed, the immune response is greater than the first time exposure. The reason is memory T cells. Same thing for B cells also, memory B cells are there. So secondary immune response is greater than primary immune response. I will come to the final topic discussion, cytotoxic T cell. Helper T cell will take antigen coming from MHC2, and MHC2 is usually concerned with antigen coming from outside into the antigen presenting cell, that is exogenous antigen. MHC1 is usually considering the antigen already present inside or broken down from the virus which is already being captured by the cell which is going to present the antigen. So this point I want you to stress. MHC1 endogenous antigen, MHC2 antigen coming from outside but entering into the cell. So please note that point, very important. Yeah, now we will go for MCQs. Autocrine stimulation of helper T cell is due to something is coming from helper. I will draw here. Something coming from helper T cell, it again comes back. Yes, what is this? MCQ, I want you to write it in the comment box. MCQ 2, my question is MHC2, which is going to be connected with helper T cell, or helper T cells is going to fight against exogenous antigen, antigen coming from outside, or endogenous antigen, antigen already present within the cell, it is not coming from outside. What could be the answer? Option 1 or option 2? So these two MCQs you please answer in the comment section. Thank you. We will meet in the next video. If you are not subscribed to our channel, Physiology Lectures by Dr. Sindhil Kumar, Dr. Sen Physiology, please subscribe to our channel. And if you want any videos to be put, please mention to our channel, mention us in the comment section that videos which you want to make. Any doubts you can try to ask in the comment section. I am ready to try to answer your queries. Feedback is also welcome. Please share this very important video on immunity. This will be definitely useful, and exam point of view it is very useful, and this is not only for first year, it is continuing lifelong reading journey. Hope this video is very useful for you. We will meet in the next video. Thank you. Happy learning.